can you guys first of all just introduce yourself a bit and also the company, also Clean?
I'm the founding CEO of Clene. Mark Mortenson, our founder and chief science officer, who will introduce himself in a second, hired me 13 years ago. And Mark and I have been together building a company around a very interesting thesis, which you're about to learn more about. Our focus is neurodegenerative disease, and in particular, ALS, with an asset soon going in front of the U.S. Food and Drug Administration for the consideration of the possibility of an accelerated approval, that is, a commercialization possibility that could occur in 27. But we'll explain that in a second. My background is 34 years in pharmaceutical drug development and commercialization.
So thanks for chatting with us. Looking forward to talking. My background is one where I simultaneously got a ceramic engineering degree and a physics degree at the same time in a 4-year time period. I now look back on that and say that's one of the most interesting things that ever happened to me, because on the physics side, I was being told there's no such thing as matter. Electrons don't exist till you measure them. They're particles and waves, which is which, and it's like all theoretical and math. And then I walk across the campus and I get into my lab or my lecture, and I'm learning how to make dielectric wafers and chips, and learning how to control the electrons by doping and changing the bands. And so what a great contradiction, right? We've got physics of the possible, and I'm making stuff even that I shouldn't even be able to be making. So it was kind of fun. Then I got a master's in material science at Penn State, and that was all to help me get into law school to become a patent attorney, because I wanted to be near invention. I was too lazy is not the right word. I was too anxious to just invent things myself. I wanted to be around lots and lots of people, cutting edge of technology. And so I'd figured out that was a great thing to be. So that's how I got started in this.
Perfect. Thanks for that. So ALS. Rob, you briefly mentioned it. ALS is, of course, a disease where motor neurons progressively fail and die. So, what is the link, I guess, between that disease process and the mechanism that you're trying to fix? So, in other words, what problem inside the neuron are you trying to fix?
So ALS has been a very difficult disease to manage. In the United States, it was commonly known as Lou Gehrig's disease because this famous baseball player contracted ALS in the prime of his baseball career. This was in the 1930s. Unfortunately, Lou Gehrig passed, but the American public, who adored and idolized Lou Gehrig, watched this disease cripple him for the first, time that really it hit the map for most people. ALS, amyotrophic lateral sclerosis, as we call it, is a disease where the neurons die. And the neurons that control everything that we functionally do with all of our muscles, our ability to talk, to walk, to breathe, to feed ourselves, to chew, all of that disappears. And it happens progressively. There are an estimated 35,000 ALS patients in America, and their survival is unfortunately devastating, with many patients passing away in 2 to 5 years. There remains no cure. There is a very bright spot, however, for 1% of ALS patients, and this is going to come up later. The company Biogen, the pharmaceutical company Biogen, has wisely developed a gene replacement therapy, a biologic that can be infused and replaces a missing gene. In a small subset of ALS individuals, about 1 to 2% of total ALS, that gene can be replaced because the only reason they have ALS is they're missing the gene. And the FDA approved this drug, despite this drug missing its primary, secondary, and exploratory endpoints. The FDA approved this drug a few years back based upon the reduction of neurofilament light, which is a biomarker in ALS. We're going to come back to this theme. But back to our asset, for the rest of ALS, in other words, the 98, 99% of everything else, it's called heterogeneous sporadic ALS. And what is happening is the neuron is under, by the disease, a devastating attack. The neuron dies. And so what our asset is doing is enabling a bioenergetic metabolite response back to the failing neuron, giving the neuron the energy it needs to fight back against the disease. Now let's have Mark talk a little bit more about this energy replacement theory that CNMAU8, I'm holding here in my hand. This is the asset that patients drink orally, is accomplishing. Mark?
So that's a good place to start. So physical part of it, this is mostly water. And it's got the color of Merlot wine because the active ingredients in it are trillions of our nanocrystals. To date, we don't require any injections. We don't require anything other than morning drinking, and it tastes just like water, smooth, like Evian water. What's different is nobody's ever decided to try to make a nanocrystal a therapeutic. And so these trillions of nanocrystals that are in here were specifically designed to do certain things. And let me first contrast what they don't do. So almost every drug you've heard to date has been intended to target a receptor or a particular protein, right? So a lock and key mechanism, turn something on, turn something off. Statins block a specific enzyme for making cholesterol. Antibiotics block a specific enzyme to keep the bacterium alive. Cancer drugs go after targets. This doesn't go after any target at all. When you drink this, it gets into your stomach, gets into your bloodstream, and gets a biological identity that's called its protein corona. The protein corona is primarily serum albumin, and serum albumin as a biological identity directs our nanocrystals to just about all sites of inflammation. So all neurodegenerative diseases, you probably know that, have inflammatory effects with them. So do a number of other physical problems. So our nanocrystals find their ways to these sites of inflammation. Then what happens is pretty interesting. Rob rightfully said, neurons demand a lot of energy. The mitochondria have to make that energy in the form of ATP. There's a lot of production of ATP. Effectively, our body weight of ATP is manufactured every single day by the mitochondria, with the neurons being the highest energy component. And so something to start with there that's grounded in the best science we have. So this is one of the things we proved out first. And this is our preclinical evidence of how this works. And so this is one of the important chemicals in the electron transport chain and elsewhere. But if you look at it, complex one. And what has to happen is NADH needs to be converted into NAD+. That's an oxidation reaction. And what we've shown you here is the optical density, how much NADH is present at time t equals 0. And then that's the molar amount of NADH in a test tube in a spectrometer. And this is the concentration of the gold nanocrystals. This is a lot higher here than what's in the test tube, but that's the concentration of gold nanocrystals in the test tube. And then as a function of time, this arrow here, we watch the NADH peak disappear. So it's gone. Over here, correspondingly, that's time t equals 0 for NAD+, and that's the exact same time point there as there. And so the amount of NADH that's been converted to NAD+ is a one-to-one comparison. So the world said, Mark, what is that reaction? I said, it's a catalyst reaction. So I'm a solid state physicist. So I learned how to make dielectric chips. I learned how to make all kinds of different wafers. Over here is a catalyst reaction. The black is the lowest ppm and the blue is the highest ppm. And this is time. And so you watch the NADH absorbance peak. See, it goes away, get less and less, like right here. As a function of composition, the NADH is changed into NAD+. The only thing that's happened there is the gold nanocrystals are present in the test tube to allow that reaction to occur. After we had done this, we did this as well. This was our most meaningful experiment. This is when Rob joined. We were doing this just when Rob was joining the company. And what you're looking at here is the cross-section of a mouse brain. These are neurons, the lighter part, so they're looking right at you. The darker part is the myelin around them. So this is the corpus callosum, and this is a high-density area, and you can see this is what normal looks like. We took 6,000 TEM photomicrographs. I can assure you, this is what normal looks like. Now we made it abnormal. What we did is we compromised the ability of the electron transport chain to make ATP. That's what we did. We put a compromising additive in the feed of the mice. They ate it, and within 2 weeks, this damage has occurred. There's the same magnification. There's swollen neurons, there's neurons without myelin, and it looks quite different than this. 3 weeks later, after the mice were drinking this version, Actually, sorry, in this case, they were actually getting an ad libitum, they were getting an injection, so gavage injection. And this treatment here, you might say, hey, it looks like it even is a little bit darker. You are looking at remyelination of the damaged corpus callosum due to the mice being gavaged a small amount of this every day for 3 weeks. Okay, so I wanted you to see that, to kind of to ground you into understanding how important it is for this to turn NADH into NAD plus. Now, the fun stuff, if I may, this is where Rob was baiting me a little bit. What's going on here is a physics phenomena. This is a catalytic reaction. These are metallic nanoparticles. If you get into a car and drive it today, there's something in there that's called a catalytic converter. Those are platinum nanocrystals that are in the catalytic converter. Those platinum nanocrystals stay there. They don't go away. They're not ingested. They're not destroyed. They just help make the CO to CO2 reaction go faster. So we're not pumping carbon monoxide into the environment or kill you. Carbon monoxide poisoning. Likewise, in our environment in a biologic, our nanocrystals are present in and around the mitochondria. We designed our nanocrystals, and if I can just go backwards, and we designed our nanocrystals so that they're highly faceted. And there's a word about plasmons that are here. This sounds like it's a little wonky, but actually you already know this and you just don't know you know it. When we were in the testing period for COVID, those little test kits that you would get, and you'd swab your nose and you'd then put it in and you'd wait for a pink line to appear. The vast majority of those COVID test kits used spherical gold nanocrystals as the indicator of whether you had COVID or not. The way that worked is there were various proteins put onto the surface of the spherical gold nanocrystals. If the spike protein was there, they bound, and then when they flowed along that pathway, they got caught up in a strip. And why did they turn pink? Because the resonant wavelength of these small gold nanocrystals took out green. They absorbed green out of normal light, and what's left is pink. So the pink line is the lack of green being in when you see it because it's been absorbed. That is localized surface plasmon resonance. I want to just let you know it's been used by a lot of different people for a lot of different things, but no one has ever used it like we did. So we understood that in the OXPHOS mechanisms of a mitochondrium, there were certain energy levels where the electrons were, right? If you look at the electron in complex 3 going to complex 4 from the oxidase to the reductase mode, there actually was a spectral signature that was there that told us something about that. Probably that's important in 2 billion years worth of biology. It's likely that spectral signal might be used by something. Turns out that spectral signature, the 520 nanometer emission, was the same frequency that was causing your COVID test strip to go pink. What I understood at that moment in time was, wow, I've seen catalytic reactions like this before. Let me make a little bit of a guess here that matters. And we set out to make highly faceted, clean surface nanocrystals. There's a whole reason that I needed a clean surface. Number one, everything that existed to date was reduction chemistry. And in 2010, the first review articles were put out on that. And the first review article said, of the 220 articles I've reviewed to date, I can't tell if a 2 nanometer is toxic and a 3 nanometer is not, a 4 nanometer is toxic and a 5 nanometer is not. It was a surface chemistry that was causing toxicity. The gold nanocrystal per se is not toxic at all. It was residual surface chemistry. We'd figured that out, and we had to invent something that would make clean surfaces, but also this representative cartoon highly faceted, because we needed to make hot spots. Just like in your catalytic converter, those are not spheres of platinum. They're jagged. They've got edges, and it makes these hot spots. Plasmons are nothing more than the electrons on the surface of a 13 nanometer crystal sloshing back and forth due to an incident electric field component of electromagnetic wave. The same thing that's causing the pink in the COVID test. We eat up the same, and then we make the field at these points easily 1,000 times stronger than the incident wave. These electrons that are bouncing back and forth in the forms of plasmons, they're the same energy levels of the electron transport chain. That's like the take-home message. We designed something to interface with the mitochondria so that for free, you could change NADH into NAD plus and more. And there's some really fun cutting edge speculation. I can go further, but that's probably a good place to stop, just to ground you to say this is based in quantum physics, because the quantum energy we're dealing with here is an electron, and we're taking the electric field of an electromagnetic wave and making those electrons concentrate as they slosh back and forward. That COVID test kit would have a concentrated field of maybe 3 to 5 times the incoming field. This point right here is 1,000 times more concentrated than the incoming electromagnetic field. That's what's causing the catalytic reaction. That's what's allowing NADH to automatically become NAD plus.
First of all, thank you very much for also sharing the slides. I think it was very useful. And so just to clarify, it's intended to be disease modifying rather than a symptomatic therapy. So something patients would take early on to slow progression.
That's the perfect question. And the problem in the field right now, if I can jump to the Technicolor dream, is those poor patients that come in with a wobble, a shake, whatever it might be. It may take 18 months to get a diagnosis before the healthcare system starts to give you what you need, or maybe not even pay for it at all. Because this is a mitochondrial dysfunction, getting to them earlier before there's serious neuronal damage is a big deal.
That was the point I was going to make simply, is that the failing neuron needs as much energy as it can receive in order to take care of its own housekeeping. And so our drug is indeed meant to be prescribed as early as possible, is designed to be taken chronically. And we now have individuals with ALS that have been taking our drug for nearing on 6.5+ years in a row. And the reason they're doing so well in a disease that's uniformly fatal in 2 to 5 years is because these are individuals that came on CNM-Au8, again, our drug taken by mouth orally, early in their disease progress. That said, even people who are well into their disease progress, if they're a neurofilament responder, that is, the drug is affecting the energy in the neuron to continue to take care of its housekeeping, even as the body is failing the individual, we see many benefits in that respect as well. Not as well, and not always, but there are responders even still latent to the disease.
All right. You guys mentioned it before. The regulatory path is ongoing. So where do things currently stand with FDA?
So we have presently completed 2 critical studies in ALS, the HEALEY study and the RESCUE study. The HEALEY is a very important study that is a platform program that Dr. Merit Sutkovich and about 50+ centers of excellence run, and 8 different drugs have come through it so far. RESCUE was a proof of concept study that we did early on in Australia. We have actually taken all this data, and though we missed the primary endpoint, what we did achieve in this study was actually quite material. We achieved the secondary of survival, listed here in these green check marks, the exploratory of clinical worsening, and also neurofilament reduction, and did this very safely. This is the secondary from HEALEY on survival. So we had a P04 benefit of a 94% risk reduction at 6 months on survival. We saw in the exploratory a P01 in RESCUE and a P03 in HEALEY on delayed time to clinical worsening. So we were on the 2 exploratories in 2 separate studies, statistically significant in both cases with a 70+% risk reduction. And in this case, just to level set your readers, clinical worsening in ALS is defined as whether or not I die, whether or not I can no longer breathe, and so I need a tracheostomy and mechanical breath to support me through a machine, or whether I need a gastrostomy tube because I can't swallow food or water. And then very importantly for the FDA said to us, look, you've got this data, but we want to focus on neurofilament light. So in that respect, neurofilament is basically release of a cytoskeletal protein remainder that is a debris trail, so to speak, that enters the bloodstream as the neuron dies. So in ALS, when the neuron is attacked, and the neuron disintegrates, it leaves behind evidence of that disintegration, and we can measure it in the bloodstream. And the reason why this is very relevant is because CLMAU8, our drug, is the first drug studied in heterogeneous sporadic ALS, so that is the 98% that are non-genetically compromised, to see a statistically significant pre-specified benefit in a phase 2 clinical study. And we saw that correlate with concordance with other biomarkers as well. And so what has happened is, because those individuals who have a neurofilament drop, they're the ones that we're seeing the best survival. We've submitted all this data to the FDA. We've had 5 meetings with the FDA in the past 18 months. And the most recent meeting was only in later March. And we announced it a week ago, Monday. And the FDA basically said, look, we think we're prepared now to accept a new drug application. You submit that, and let's discuss this data and send it into review so that we can really get under the hood of your data as is required in the U.S. regulatory back and forth and let you know what we think about this data. But the key is to use neurofilament as a surrogate for accelerated approval with the addition of the clinical response. And in this case, the clinical response is survival.
Let me add something to that, Rob and Federico. So I'll share my screen again, because I think this is as important, and it's showing the regulatory flexibility of the FDA. So we had the largest compassionate use in ALS ever. And the NIH, National Institute of Health, gave us a $45 million grant to go out and provide this drug to people who were much more advanced than any clinical trial, including ours. You know, they wouldn't qualify for it. So if you look down here at the beginning, we have 258 people in this compassionate use, but we tracked them also their neurofilament light biomarker. So this wasn't just a compassionate use, it's also convenient for the patients, but we're also tracking the biomarker. And we're out to 4 years. 8 people have gotten out that far. There's a lot more still coming along because this is still ongoing here. But what we did is we matched our drug response to covariate controls, 2 different ones. It was called Answer ALS in this particular case. And we're seeing the same survival signal, and we saw commensurate in scope changes in neurofilament light. And most importantly, the FDA said, let's together, before you analyze this, figure out an analytic technique where you can look at this in a blinded way, and we'll agree with that approach. And then if it gives you good data, we'd like you to include that in the new drug application that Rob just mentioned. And so that is a really good sign that the FDA is quite interested. And we were delighted to see these people much further along in their progression show the same changes relative to covariate matched data controls and NFL changes that were almost identical. So that was heartwarming. And here's the FDA, because we didn't hit our primary endpoint. So stats people say, thumbs down, go do it again. But it's other folks in the FDA saying, wait a minute, there's a survival signal here. And clearly, if survival was our primary endpoint, this is an approved drug. There's a survival signal here. Let's see if we can build a story. So this is still strict evidence-based approval, but showing regulatory flexibility to, you missed your primary endpoint, but here's the accelerated approval pathway. Connect the dots for us to your NFL biomarkers and any other clinical events with survival, and we'll be willing to review that for you.
In fact, adding on to that for just a second, the FDA requires in the notion of an accelerated approval, a confirmatory phase 3 that would commence. And Clene has already announced that phase 3 will start in the coming months. Our design is a phase 3 with survival, overall survival as the primary endpoint. And this is actually a remarkable step forward in ALS. To actually have the clinical primary endpoint be overall survival is a very relevant discussion. But the reason that we've done that is, as we've already hinted here, and there's many more slides we could show, is that we've seen survival in lots of different assessments, including against concurrent controls from the HEALEY study. And the agency has seen this. Overall survival is not an option of approval in the FDA statute for accelerated approval, but a biomarker is. And so that's why the FDA has done exactly as we've expressed in our previous press release, is they basically said that our proposed data may be capable of supporting and the review of a new drug application. And it's critical that since neurofilament light, this biomarker we've showed you, it could serve as what they call a reasonably likely surrogate endpoint to support the accelerated approval. So what we've done now is prepare all of this data. We're submitting it in a new drug application later on this year to support exactly this, the connection between the magnitude of reduction in neurofilament and the clinical benefit, survival plus.
I also wanted to briefly talk about safety because this is a gold nanocrystal suspension. Some viewers may wonder whether gold accumulates perhaps in the body, creates toxicity concerns. What would you say about that?
Let me show a slide, and while let me first answer your first question, and then Mark can speak about the nanoparticles in the body. So we now have over 1,100 collective participant years of exposure to CNM-Au8 across multiple programs. We have duration that's now numbering, I mentioned a minute ago, 6.5 years. We have over 900 patients treated. We have not a single, again, I'll triplicate this for emphasis, not a single related serious adverse event that has been attributed by any clinician to CNM-Au8. The agency was quite worried at the beginning that a nanoparticle that was taken by mouth systemically and makes its way into the tissues of the body, including the mitochondria, would there be any concern about accumulation? So they had us do extensive animal work across multiple species and across multiple doses in our phase 1 human work. And we've seen to date, knock on our collective wood here, not a single serious adverse event. We have some side effects of nausea and headache, but they're transient. They're not related to longer term use of the drug. Mark?
Two ways to think about this. So catalysts only promote, this particular catalyst in particular, only promote naturally occurring effects. So if you were to go back mentally to those neurons that we showed of the mice, we tried to see if we got any Frankenstein effects at all, right? Is there anything that's abnormal by having this? I knew what the answer was going to be because a catalyst automatically seeks homeostasis in a system. It doesn't push something past the point, it tries to bring it to that point for homeostasis. In our Parkinson's MRI studies, so we had both Parkinson's and MRI patients, because an early on question that Rob said we've got to address this is there target engagement in the brain? We had patients have magnetic resonance spectroscopy, that high Tesla value, not the low Tesla values that we get when we go to look at our knee, but 7 Tesla. And we looked at the phosphorus components like the ATP, phosphorus in human brains. And in 90 days, we saw differences where people who were low went back up. We've not had a single serious adverse event because the catalyst is seeking homeostasis. Just like the platinum catalyst sitting in your car, it does nothing until there's an exhaust flow. It's just sitting there. As soon as there's an exhaust flow and there's carbon monoxide on this side, it goes, okay, grab some oxygen, make CO2. The gold, if it gets into a system like being perfectly healthy, it doesn't have any jobs to do. There's nothing for it to catalyze. So it just passes out the body.
Okay, I think I have 1 or 2 questions left. First of all, what about the manufacturing? It can be, I guess, a tricky one, particle size, surface chemistry, consistency as well. So how difficult is it to manufacture CNM-Au8?
So this is an older, early generation version. This is the clean room, one of our clean rooms sitting right behind me right now. And what's up here is an apparatus where there's a series of gold wires, and these pairs of gold wires are in a continuously flowing water unit. We just call them troughs. And what happens is, this is the output of the trough, this is the input here. What happens is we strip off ions of gold one at a time. We add an atom of gold to that, we add an electron to that ion of gold to make it an atom. And the fields that are created, electric and magnetic fields right around there, and the flow rate of the water All of that is controlled by a very sophisticated system here. Every drop of water we make every day is the same. There has to be a continuous production period. You can't do this in a batch process, because in a batch process, everything's changing every second. Right here, the same thing happens every single time at that set of electrodes, that set of electrodes. And this is set up so that we put about 60,000 atoms of gold into every one of the trillion of nanocrystals that's in here. They are highly faceted. They're in three shapes intentionally. It's called, just think of a pyramid on top of a pyramid, upside down, base to base. Think of a hexagon, like the design on a soccer ball, and think of triangular plates, one that looks like a triangle this way and flat kind of that way. We make those three shapes intentionally. They're in a very tight particle size distribution. We didn't want just one exact size. We wanted something that ranged from 9 to 15, but it's the average on 13. There's reasons for that biologically that we wanted that. And every single drop that we make is identical to the last drop. What's happened here is, while these recipes, we've got 100 recipes for making various nanocrystal suspensions that we shared with the world. What happens is, when this is collected and these tubes here lead to large, many thousands of liters collection tanks, then what happens after that, we've kept proprietary. It turns out it's no small feat. It took us years to change the first 7 gallons into this. So we effectively squeeze the water out of this light pink solution that's eating up 520 nanometer green wavelength to get this darker color. It's just this exact same material, but a lot of water has been squeezed out of there. So for example, this is more than 70 times more concentrated than what you see right there. These nanocrystals are highly reactive surfaces. We invented them to be that way. We invented a way to keep them suspended. The reason I mention that proudly is everyone before us was so happy with the different materials they added to the gold, the surfactants, to keep them suspended. We figured out a set of conditions where we simultaneously lyse water, and we take the OH groups of water, and that is our identity in the bottle. OHs banging off each other like negatives on a magnet, so we're able to concentrate that. As soon as you put a little bit of saliva into this, if it was just sitting there, watch it for a couple hours, it'll completely change. They're highly reactive. Get it into your body, no problem. There's no coagulation that occurs. They get into your stomach, the acid breaks stuff that are off them, and we've got clean surface gold nanocrystals with a protein corona flowing to all the inflammatory sites in your body.
I'm going to ask one last question. Yeah. How large is the market you're targeting?
Good question. So there's 35,000, as I just mentioned, ALS people. To put a beautiful surrogate in, another company launched in America a few years back, and unfortunately, their drug did not work in their confirmatory phase 3, so they had to pull their drug from the market. But within the first year, they were estimated to sell 4,000 people. So what is that? 10 to 15% of the entire market within a number of months. They were on track for that reason. And in our case, our drug is even easier to take than this other asset, given that it tastes like water and one drinks it as water when one wakes up in the morning. So the market potential is quite large. We've not mentioned it all, but Clene is also, with the same asset, same dose, preparing a phase 3 that we hope to launch next year in multiple sclerosis. That market is gigantic. There are 2.6 million multiple sclerosis people worldwide, and many of them have a cognitive or visual deficit. And that is exactly what our drug, again, it's giving energy to the failing neuron. So in MS, there's been lesions that have attacked the neuron, giving either cognitive compromise or vision compromise. It's a whole separate discussion. But what we're seeing here is a pipeline and a product. So we're launching with ALS because the obvious immediate devastation of ALS warrants our focus there first, but we envision other neurodegenerative diseases, including multiple sclerosis, and then a little bit after that, there's a very large opportunity in Parkinson's and other areas as well.
The idea is to target also broader neurodegenerative disease.
That's
Federico,
true.
the way I say that is, there's been a lot of really bright people for a very long time targeting different proteins and enzymes to turn things on or turns things off. No one has looked at the root cause upstream and said, why is the cell dying at the most fundamental level? And the answer is because of the lack of energy. every disease where there's mitochondrial dysfunction, and you'll find it's every neurodegenerative disease and more, will benefit from exactly the composition we have now. That doesn't mean we won't make better ones, but everyone will benefit from the composition we have right now, which means no phase ones if we keep the composition the same. Just go right into phase 2 and sometimes phase 3s.
Thank you for your time and attention to Clene. It's an exciting opportunity for neurodegenerative disease, and we're very grateful that the US FDA has taken some consideration for the possibility of a new drug application filing this year, which they would review for potential commercialization. And just since you're both based in Europe, the UK and I think in Italy, a note about this. So we plan to launch our phase 3 confirmatory study in Europe, as well as globally. We've already had preliminary discussions with EMEA. There's a deep market need in Europe. There's only one drug available in most European countries. And so if we can conclude the phase 3 study successfully and take our drug, it could be potentially the next asset in Europe as well.
That sounds exciting. Rob, thank you for that.